Conception, a view from the cow. The fertilization of a mature egg by a sperm cell marks the start of a new life. In large mammals, such as cattle and humans, the oocyte undergoes months of preparation for this event. It takes approximately 6 months for a bovine oocyte to develop from its resting storage state to that of a mature egg, which is ready to be fertilized and can direct the early cleavage divisions and initiate activation of the embryonic genome, necessary for the developing embryo to take control of its own fate. The morphological changes that are integral to oocyte growth, development and fertilization have been revealed through the application of light and electron microscopy. Small pieces of ovarian cortex or isolated oocytes and embryos are fixed and embedded in apon, and then cut into semi-thin sections for light microscopy and ultra-thin sections for electron microscopy. Sectioning is performed by means of an ultramicrotome. Transmission electron microscopy allows us to visualize the tiny organelles within the cytoplasm of the oocyte and embryo at up to 100,000 times magnification. In cattle and all other mammalian species, oocyte growth takes place in the ovaries, within follicles. Starting with activation of the resting primordial follicle and culminating with the ovulation of a matured egg, which, if fertilized, will support pronucleus formation in the zygote, then cleave to the 2, 4, 8 and 16 cell embryo and eventually form a blastocyst. Let's focus on this series of developmental stages and zoom in on oocyte growth. During this phase of development, the oocyte grows from 30 to 120 micrometers in diameter, the equivalent of a 64-fold increase in volume. Growth is accompanied by the synthesis and proliferation of various organelles. Oocyte growth commences with the activation of the resting primordial follicle. The resting primordial follicle is characterized by a flattened layer of granulosa cells, shown here in green, a large nucleus, shown here in orange, and cytoplasm, shown in yellow. Note the size of the nucleus. It occupies approximately 15% of the oocyte volume. Zooming in, first on the nucleus, we note lightly condensed chromatin distributed throughout, and an inactive nucleolus within. Looking at the cytoplasm, the organelles are accumulated in the centre of the oocyte. The oocyte's mitochondria are characterized by their round shape, and many of them contain what appears to be mitochondrial granules within. The mitochondria are associated with smooth endoplasmic reticulum. We also see clusters of ribosomes distributed throughout the cytoplasm and accumulations of microtubules. Primordial follicle activation occurs independently of gonadotrophins. KIT ligand produced by the granulosa cells binds its receptor on oocytes, triggering the phosphoinositide 3 kinase, protein kinase B cascade within oocytes. Primordial follicle activation is characterized by proliferation and restructuring of surrounding granulosa cells to form the primary follicle. The primary follicle is characterized by a complete layer of cuboidal granulosa cells. The organelles within the cytoplasm appear to have migrated towards the nucleus, while subtle changes to the chromatin and nucleolus are noticeable within the oocyte nucleus. The nucleolus is the site of ribosomal RNA transcription and eventual biogenesis of ribosomes through sequestration of ribosomal proteins. Ribosomes are composed of a larger 60S and smaller 40S subunit. The two subunits come together during messenger RNA translation in the cytoplasm. The primary function of ribosomal RNA is in protein synthesis. Thus, the presence of ribosomal transcripts may serve as a marker of transcription and protein synthesis. If we look carefully at the structure of the nucleolus, we can elaborate the cycle of nucleolus activation, transcription, and quiescence as it occurs during oocyte growth and embryo development. We will detail these changes as we sequentially address the stages of oocyte and embryo development. The nucleolus in the oocyte of the resting primordial follicle is characterized by a reticulum in which granules are embedded. The shape and size of the nucleolus is influenced by the size and number of the reticular spaces, also referred to as vacuoles. Such a nucleolus is inactive with respect to transcription and ribosome synthesis. In the primary follicle oocyte, we note the appearance of one or more fibrillar centres in close proximity to the nucleolus, marking the impending nucleolus activation. The next stage of development is the secondary follicle. Continued granulosa cell proliferation results in the formation of two or more complete layers of granulosa cells. This is a very interesting stage of follicle development in cattle. Zooming in on the interface between the granulosa cells and the oocyte, we see that the granulosa cells extend processes towards the oolemma, the oocyte membrane. We also note the formation of microvilli on the oolemma and the first signs of deposition of materials for the zona pellucida, seen here in yellow. The connection between the granulosa cell processes and the oocyte is established first by adherens or structural junctions, but ultimately communication channels in the form of gap junctions facilitating bidirectional communication between the two cell types are established. Zooming into the oocyte cytoplasm in the secondary follicle, we see the first traces of the cortical granules, which will play an important role during fertilization. Actually, the origin of the cortical granules is still enigmatic, but here we see them associated with large vesicular structures in the oocyte cytoplasm. We also see further evidence of nucleolus activation in the oocyte nucleus, with the migration of the fibrillar centres into the granular reticulum. This integration marks the formation of a functional nucleolus capable of transcription and ribosome synthesis. As the granulosa cells continue to proliferate, small fluid-filled spaces develop between them, which eventually coalesce to form the antrum, hence the term antral or tertiary follicles. In the tertiary follicle, we can appreciate how much the oocyte cytoplasm has increased in size, as the volume of this compartment is much larger relative to the oocyte nucleus than in the earlier follicle stages. Zooming in on the oocyte cytoplasm, we now find that the oocyte is furnished with most of the organelles and inclusions required for further development. Large vesicles potentially containing energy sources physically connected with mitochondria, as well as big Golgi complexes and clusters of cortical granules are seen. The granulosa cells which surround the oocyte are also referred to as cumulus cells. If we look more closely at the interface between them and the oocyte, we now see that the oocyte is completely encased in the glycoprotein shell called the zona pellucida, and we also see oocyte microvilli have become embedded in the zona pellucida. Within the nucleus, the nucleolus has reached its peak of activity with respect to transcription and ribosome synthesis, evidenced by numerous fibrillar centres interspersed throughout the granular reticulum. Antral follicle growth is marked by an accumulation of follicular fluid in the antrum, the differentiation of granulosa cells into the cumulus cells which surround and nurture the oocyte, and the mural granulosa cells which secrete the basement membrane. Theca cells align and proliferate on the outside of the basement membrane. At this stage of development, the follicle has transitioned from being gonadotrophin responsive to gonadotrophin dependent for continued growth and development. Oocyte growth continues in parallel with the follicle until the follicle reaches a diameter of 2 to 3 millimeters. At this point, oocyte growth plateaus at approximately 110 micrometers. At this stage of oocyte development, the first indications of nucleolus deactivation are observed. The fibrillar centres are marginalized to the periphery of the granular reticulum. Finally, the nucleolus compacts to an electron-dense fibrillar sphere, with a fibrillar centre attached in the form of a halo. Occasionally, electron-dense granules are associated with the nucleolus. The oocyte completes its growth period at a final diameter of between 120 and 130 micrometers. The cytoplasmic organelles disperse towards the periphery of the oocyte, and looking more closely, we see that at this stage, the mitochondria have assumed a hooded shape and the cortical granules have become located in large peripheral clusters, both characteristics of ruminant oocytes. The oocyte is now fully grown and it is competent to resume meiosis in response to an endogenous surge in luteinizing hormone, also known as the LH surge, or recovery from the ovary and submission to in vitro oocyte maturation. Oocyte maturation is essential for achieving two important missions: extrusion of half of the chromosomal content to establish a haploid female gamete, and protection against polyspermic fertilization. These events are initiated by signals triggered by the ovulatory LH surge, leading to an acute upregulation of the epidermal growth factor signaling network in mural granulosa cells. The epidermal growth factor signaling cascade is propagated from the mural granulosa cells to the cumulus cells, resulting in initiation of mural granulosa cell luteinization, closure of gap junctions, and production of an extensive extracellular matrix by cumulus cells. The resulting downregulation of the meiotic inhibitory signaling network, mediated by C-type natriuretic peptide, or CNP, and cyclic guanosine monophosphate, in mural granulosa and cumulus cells, promotes resumption of oocyte meiotic maturation. Focusing on the communication between the cumulus cells and the oocyte, there has, until the onset of oocyte maturation, been close interaction through gap junctions. Zooming in, we can clearly see how long cumulus projections traverse the zona pellucida. This intimate communication is broken following the onset of oocyte maturation. The first sign that meiosis has resumed and that the first meiotic division is progressing is the dissolution of the oocyte's nuclear envelope, referred to as oocyte nucleus or germinal vesicle breakdown. The remnants of the nuclear membrane are shown here in green. Simultaneously, the loose chromatin starts its packaging into chromosomes in order to progress to metaphase 1. Gradually, the nuclear envelope is totally dissolved into smooth endoplasmic reticulum, and during the first metaphase, the paired homologous chromosomes align in a metaphase plate. They attach to their respective centromeres by spindle fibres. The stage from germinal vesicle breakdown and chromosome compaction to the alignment of the chromosomes on the first meiotic plate is defined as pro-metaphase 1 and lasts for several hours. It can take a few attempts for microtubules to assemble into a spindle and attain the final conformation at metaphase. This phase of oocyte development is critically dependent on the tightly orchestrated translation and degradation of messenger RNAs, which were accumulated in the oocyte during the growth phase. Subsequently, meiosis progresses from metaphase 1 to anaphase 1, when the chromosomes begin separating, with one set of homologous chromosomes being extruded as waste in the first polar body extrusion during telophase 1. Once the first polar body is extruded, the oocyte is considered mature. It is at second metaphase, or metaphase 2. Zooming in on the region around the first polar body, we note the perivitelline space and the cortical granules, shown here in blue. The cortical granules have lined up beneath the olemma and are ready to expel their contents into the perivitelline space, following fusion between the oocyte and the fertilizing spermatozoon. In cattle, the mature oocyte is expelled from the ovulatory follicle approximately 24 hours after the peak of the LH surge. It will be funneled into the oviduct and transported towards waiting sperm, if the animal has been mated or inseminated. This sets the scene for fertilization. Fertilization involves several critical steps, which must be achieved in order to produce a viable embryo. First, the spermatozoon must penetrate the zona pellucida to enable fusion of the two gametes, and the chromosomes of each gamete must be segregated into their respective maternal and paternal pronuclei. First, we will focus on gamete fusion. Scanning electron microscopy allows us to visualize spermatozoa, shown here in blue, and cumulus cells in green on the surface of the zona pellucida, which has been coloured yellow. On the zona pellucida surface, the spermatozoa will undergo the acrosome reaction, exposing several enzymes which digest the zona pellucida, allowing the sperm cell to travel through the zona pellucida into the oocyte. 3 processes are necessary for successful fertilization: sperm motility, sperm capacitation, and the acrosome reaction. Calcium signaling plays a critical role in their regulation. The sperm plasma membrane undergoes marked biochemical changes during capacitation. This involves the depletion of seminal plasma proteins from the sperm membrane to reveal oocyte binding proteins on the surface of the acrosome. The zona pellucida of the oocyte consists of 3 zona pellucida proteins. The sperm plasma membrane undergoes marked biochemical changes during capacitation. This involves the depletion of seminal plasma proteins from the sperm membrane to reveal oocyte binding proteins on the surface of the acrosome. The zona pellucida of the oocyte consists of 3 zona pellucida proteins, ZP1, ZP2, and ZP3. Binding of the sperm to ZP3 initiates the sperm acrosome reaction. The acrosome reaction enables spermatozoa to penetrate the zona pellucida, and it also exposes the sperm equatorial segment. The acrosome reaction begins when the plasma membrane of the spermatozoon forms multiple fusion sites with the outer acrosomal membrane. Fusion of the 2 membranes results in the formation of many small vesicles, termed vesiculation. After vesiculation occurs, the acrosomal contents are dispersed, leaving the sperm nucleus with just the inner acrosomal membrane around it. When the spermatozoon completely penetrates the zona pellucida and reaches the perivitelline space, it settles into a bed of microvilli on the surface of the oocyte plasma membrane. The olemma fuses with the exposed spermatozoon equatorial segment, and the fertilizing spermatozoon is engulfed. The full spermatozoon, including the tail, is taken up into the oocyte as the sperm plasma membrane is gradually integrated into the olemma, while the sperm content is released into the ooplasm. Zooming in on the head of the fertilizing spermatozoon, we see that the sperm chromatin is initially completely compacted. Immediately upon gamete fusion, a cortical reaction occurs in the oocyte. Returning to the oocyte surface, we see that the cortical granules release their content into the perivitelline space by exocytosis. The enzymatic content of the cortical granules provokes hardening of the zona pellucida, blocking the entry of supernumerary spermatozoa into the oocyte. Zooming in on the spermatozoon in the ooplasm, we see that within just a few hours after gamete fusion, the chromosomes in the sperm head have started decondensing, and looking more closely, we see the migration of smooth endoplasmic reticulum towards the decondensing sperm head and the formation of nuclear envelope fragments. At this point, the sperm tail is segregated from the head and is broken down. The sperm mitochondria also undergo a similar fate, resulting in exclusive maternal inheritance of the mitochondria and the mitochondrial genome from the oocyte. Within the following hour, the decondensing chromosomes of the sperm head become surrounded by a complete nuclear envelope, and the paternal nucleus is formed. A similar process occurs with the maternal haploid chromosome compartment, resulting in the formation of the maternal pronucleus. During the next hour or two, chromosome decondensation ensues. The pronuclei swell to their characteristic spherical shape, and they are visible approximately 6 to 8 hours after ovulation. Over the next 10 hours, the pronuclei migrate towards each other. DNA is replicated during this period, and at approximately 20 hours post-ovulation, the envelopes of both pronuclei are broken down. This allows for the maternal and paternal chromosomes to come in contact and occupy the central ooplasm during a process referred to as synkaryosis. Fertilization has taken place, and initial embryonic development commences with the first cleavage to the two-cell stage. Each blastomere has now received a full copy of both the maternal and paternal genome. Early embryo development, that is, the first 4 cleavage divisions, is dependent on transcripts and proteins accumulated during oocyte growth. The first cleavage division occurs between 24 and 36 hours after ovulation. There is evidence to suggest that early cleaving embryos are more developmentally competent than their later cleaving counterparts. Subsequently, second and third cleavage divisions commence. Each cleavage division sees a doubling of the cell number and takes approximately 24 hours. Thus, we expect to find an 8-cell embryo in the oviduct at approximately 72 hours post-ovulation. During these early cleavage divisions, we see the gradual activation of the embryonic genome. While the pronuclei, as well as the nuclei of the 2- and 4-cell embryo, do not contain active nucleoli, they contain so-called nucleolus precursor bodies, which to a high degree share morphology with the electron-dense fibrillar sphere of the fully grown oocyte. At the 8-cell stage, however, the nucleolus precursor bodies become vacuolized, and fibrillar centers connected to chromatin and surrounded by a dense fibrillar component, which consists of newly transcribed ribosomal RNA, become embedded in the periphery of the precursor bodies. These events mark the reactivation of the nucleoli with respect to transcription and ribosome synthesis. Finally, fully functional ribosome synthesizing nucleoli appear in the 8-cell nuclei. Such nucleoli consist of fibrillar centers, a dense fibrillar component, and a granular component surrounded by chromatin. The location of the ribosomal DNA, the ribosomal RNA genes, are reported to encircle the fibrillar centers to allow transcription of the ribosomal DNA to proceed. Hence, nucleolus function can tentatively be envisioned with the ribosomal RNA transcription taking place at the periphery of the fibrillar centers, and the newly transcribed ribosomal RNA accumulating as dense fibrillar component around the fibrillar centers, offering itself for processing and packing with proteins to form the ribosomal subunits that make up the granular component. Finally, the ribosomal subunits are exported to the cytoplasm through the pores of the nuclear envelope, where they engage in translation and protein synthesis. The developing embryos leave the oviduct at this point and move into the uterine horn. In cattle, pregnancies are generally carried in the uterine horn, ipsilateral to the site of ovulation and corpus luteum formation. Subsequent cleavages lead to the formation of the morula, consisting of 32 to 64 cells. The morula compacts due to the formation of an outer cell layer, the trophoblast, shown here in blue, where the cells tightly adhere to each other. The trophoblasts surround the inner cell mass. The inner cell mass is pluripotent and will develop into the embryo proper. Blastulation occurs at around day 7. Fluid is accumulated in the blastocyst cavity, resulting in positioning of the inner cell mass at one pole of the embryo. The formation of the blastocyst on day 7 marks an important milestone in embryo development in cattle. The embryo is now in control of its own fate, and it is also the stage which can be applied to reproductive biotechnologies, such as embryo transfer and embryo freezing for later transfer.